Hello, my project is on investigating the role of a ubiquitin B knockout in amyloid aggregation using a novel H9C2-derived cardiomyocyte model of transthyretin amyloidosis to address a novel preventative treatment via ubiquitin. Initially, I was inspired to pursue this research due to watching a patient with the disease debate physician-assisted suicide firsthand due to the mere cost of treatment. To mitigate this, I was motivated to evaluate ubiquitin B's biomarker potential. Transthyretin amyloidosis, or ATTR for short, primarily affects the left ventricle of the heart, which is responsible for pumping blood throughout the body. The disease results from the aggregation of misfolded transthyretin proteins, leading to amyloid deposits. It is also severely underdiagnosed because it lacks specific cardiac symptoms that differentiate it from general cardiac failure. Current treatments on the market, like tafamidis, are transthyretin stabilizers, which are extremely costly, ranging to up to $10,000 a year, even with insurance. Or the other plan being heart and liver transplants, which are both medically invasive and specific to the patient. The pathology I looked at stems from the liver, which creates the transthyretin tetramer, which then disassociates, misfolds into amyloid fibrils, and accumulates in the heart to cause restrictive cardiomyopathy. To combat this, I analyzed the ubiquitin proteasome system, which focuses on cleaving these misfolded proteins. It works with ubiquitin, E1, E2, and E3 ligases to first tag the misfolded protein and then degrade it via proteasome. I hypothesized a relationship because both of the pathways focus on misfolded proteins. My first research question and hypothesis focused on whether the knockout of ubiquitin B affected amyloid aggregation and cell viability via cell cleaving functions. My second one investigated ubiquitin B's biomarker potential for diagnosing ATTR by comparing healthy and ATTR model cardiomyocytes. The purpose of the study was to investigate the role of ubiquitin B as a potential biomarker to increase earlier diagnosis in three distinct steps. So first, evaluate ubiquitin B's potential via immunohistochemistry and Western blotting. Secondly, to model ATTR pathology in cells via cardiomyocytes and B122I human recombinant protein supplementation. And finally, to look at the ubiquitin B siRNA transfection and compare cell viability via the MTC assay. My first assay on immunohistochemistry revealed that when comparing the healthy control and ATTR ubiquitin values, ATTR tissues had 144% more ubiquitin and 86% higher staining intensity compared to the controls with a p-value less than 0.05. This suggests that ubiquitin accumulation may be a compensatory response to amyloid stress, reinforcing its diagnostic potential. Western blotting also quantified ubiquitin levels, revealing a significant increase in heart failure samples compared to ATTR samples in cardiac tissue. This is likely because of the increase in ubiquitin via heart failure due to the decrease in alive cells in ATTR. This could also help cardiologists with differentiating between general cardiac failure and ATTR as a whole. Secondly, there is no significant difference between the ATTR cardiac samples and the healthy control. This is likely due to the amount of dead cells in the ATTR cardiac tissue, which causes an unlocalized quantification of ubiquitin, thus decreasing overall levels. The methodology that I looked at in vitro modeling focused on transitioning the cardiomyocytes into UBB cardiomyocytes from H9C2 myoblasts. Here you can see the four distinct phenotypes. They all had statistically significant values that confirmed their phenotypic ability, and the red circles indicate amyloidogenic hotspots. My last assay was an MTC assay on cytotoxicity evaluation, and we can see here that comparing cardiomyocytes to TTR cardiomyocytes, the transthyretin protein did work by decreasing cell viability to model the disease, and comparing that to the UBB siRNA, ubiquitin may have a potential protective role. The hypothesis was supported, and ubiquitin could be a potential biomarker. The experiment opens avenue to decreasing costly therapies by 25 times, safeguarding cardiac health, and establishing a novel economical model of the disease to model it in cells. Comparing this to industry standards, all current treatments are significantly more costly than the novel UBB biomarker monitoring. The study is short-term, so it does not capture the full effects of a long-term study. I hope to explore proteasome in the future, measure UBB via qPCR, and model the disease biologically through Congo red staining. Thank you.